So today's topic is fertilization. In my previous lectures, I have told you about the oogenesis and the spermatogenesis. Once that is done, next process is the process of fertilization. So what is fertilization? Fertilization is basically the fusion of male and female gametes to form a zygote. Now the sperm that is released inside the female genital tract, it needs to be matured in order to fertilize the egg. So what processes take place in a sperm so that it is able to fertilize an egg are the capacitation process and the acrosomal reaction. Now this picture very well depicts the process of capacitation. This is an uncapacitated sperm and this is a capacitated sperm. So what happens is incapacitation, the proteins on the surface of the sperm, they are removed. And the capacitation basically is a period of conditioning that occurs in the female genital tract. It lasts as long as 7 hours. If a sperm reaches the egg earlier than this time duration, it is of no use because it won't be able to fertilize the egg. So it needs to be capacitated in order to fertilize an egg. So what is capacitation? It is a period of conditioning. Yeah, and in it what happens is that the seminal proteins and the glycoprotein that is removed, and the sperm becomes active, and all this process is occurring in the uterus and the uterine tube. Next is the process of acrosomal reaction. Once a sperm is capacitated, it undergoes acrosomal reaction. The capacitated sperm, it contacts the oocyte, and a few molecular changes occur. What those changes are that the oocyte has integrins which bind with disintegrants on the sperm. It leads to the release of enzymes, the hyaluronidase and the acrosine. As I've told you earlier, fertilization is the process of fusion of an egg and a sperm, and this most often occurs in the ampulla of the uterine tube, that is the most wide part of the uterine tube. Now, in this picture, we can see these outermost cells are the corona radiata. This gray lining is the zona pellucida, and in this is the oocyte having the cytoplasm and this nucleus. What are the various phases of fertilization? First of all, the sperms, they have to pass through the corona radiata, that is the outermost layer. Then they will penetrate the zona pellucida. After that, the fusion of plasma membrane of the sperm and the oocyte occurs. Next, the completion of meiosis II of oocyte occurs, that occurs after the process of fertilization. Until that, it is arrested. Finally, there is formation of the male and female pronuclei which will fuse and a zygote will be formed. These are briefly the phases of fertilization. So first of all, we'll discuss briefly the passage of sperm through the corona radiata. This is the layer of corona radiata. The sperm that is capacitated, it will penetrate the corona radiata by multiple factors which are release of the hyaluronidase, and it depends on the sperm motility. In this picture, we can see multiple sperms that are trying to enter across the corona radiata, but only one sperm is able to finally reach inside of the oocyte. The zona pellucida, I've told you in my previous lectures that it is a layer of glycoprotein, and this is released by the oocytes inside and the granulosa cells on the outside. Between the granulosa cells and the oocyte is a layer of glycoprotein, this reddish structure, which is the zona pellucida. The zona pellucida has 4 proteins, out of which the ZP3 protein is responsible for binding of the sperm. Next step is the penetration of zona pellucida. What happens is that the various enzymes like esterases, acrosine, neuraminidase, these enzymes help the sperm penetrate the zona pellucida. Then there is a zona reaction. This is very important. What happens is that the oocyte, it releases various cortical granules inside the perivitelline space. What do? They act as lysosomal enzymes. They prevent multiple sperms from penetrating the zona pellucida. So it prevents basically the polyspermy. It prevents many sperms from entering into the pellucida. It allows only one sperm to reach the zona pellucida. Next step is the fusion of plasma membranes. What happens is during acrosomal reaction and capacitation, the plasma membrane of the sperm is removed. So only the head and tail of the sperm enters the oocyte and fuses with the plasma membrane of the oocyte. In this picture, we can see that this is the capacitated sperm. It releases granules, which tries to bind and then penetrate the zona pellucida. It finally enters into the crosses through the zona pellucida. The plasma membrane of the sperm is left behind. Only the nucleus enters. It binds with the oocyte nucleus and forms a zygote. Next is after fusion of the male and female pronuclei, there is completion of the meiosis II of oocyte, and it leads to activation of the egg and completion of the meiosis II and formation of a mature oocyte and the secondary polar body. The male and female pronuclei are formed and they fuse to form a zygote. So now an ootid is formed, which is a zygote containing the male and female pronuclei, but it is still immature. In this picture, we can see that this is nucleus of the ovum. It binds with nucleus with the sperm and forms a zygote. So the results of fertilization are variable. It restores the diploid number of chromosomes, the variation of human species. The chromosomal sex of the embryo is determined at this time. Metabolic activation of ootid occurs, and finally initiation of cleavage happens. So this is all about fertilization. You can ask me any questions in the taskbar.